All right, chapter 12 is on DNA replication and recombination. Okay, DNA replication and recombination. So, this young boy that you're looking at, his name is Ryan, and Ryan was born with a genetic condition known as Meyer-Gorlin syndrome. And Meyer-Gorlin syndrome results in an individual that has deformities. Usually they are small for their actual gestational age. They usually have either no kneecaps, they have bone abnormalities, feeding issues. They can be intellectually normal, but have a normal lifespan, but have abnormal development, usually due to bone abnormalities and bone formation. The etiology of Meyer-Gorlin syndrome has been tied back to DNA replication, hence why it is a part of this chapter. Okay, it is tied back to a flaw in DNA replication, and what one of the important factors of this issue is in children, they experience a reduction in the growth development, prenatal development, I should say. Okay, and this is a key process to the replication machinery in regards to licensing factors that occur early in the cell cycle process. So I want to give just a physical or tangible association with what we're getting ready to talk about, DNA replication, and how this can affect humans. So when we're talking about, before we can talk about DNA replication, we have to talk about the genetic information and the accuracy of it. Okay, replication is the actual processing or duplication of genetic material. However, this genetic material that is copied before a cell divides has to be extremely accurate. Okay, but we know that there are errors that occur. It's actually believed that there is one error that occurs per million base pairs, which leads to about 6,400 mistakes every time a cell divides, which, when you think about it, could be very detrimental. I want you to keep in the back of your mind, if this is the case, then how are these repairs, how are these errors repaired, or why do they not show up all the time in every cell that is potentially containing these errors? One of the reasons or contributing factors to these genetic mutations is that replication is occurring at a fast rate. And so because replication is occurring at a fast rate, it's inevitable that you're going to have mistakes that are incorporated into the growing DNA. It's occurring at such a fast rate, this is about 1,000 nucleotides per second. Okay, 1,000 nucleotides per second. So, in regards to DNA replication, the way in which it replicates was not known for a long time. So there were different models that were proposed to determine how DNA is replicated. Now, again, what is DNA replication? It is the copying of genetic material. But understanding the intricacies of how this occurs was not fully understood. So there were three models that were proposed. You have the conservative replicative model, the dispersive replication model, and the semi-conservative replication model. So we'll talk about these three different models. Okay, so as you see here, the first replication model, which is the conservative replication model, ultimately suggests that you have the parental DNA that is used as a template to make daughter DNA. Okay, as you see here in blue, this is the parental DNA. Okay, once the parental DNA in blue is used, it still comes back together, as you see here, or reanneals, and then the new DNA comes together. So they're actually, the parental DNA is only used as a template, but it goes back to it being a double helix within itself, and the newly formed DNA comes together. In this dispersive replication model that was proposed, it suggests that you have this DNA that is used as a template, the parental DNA that's used as a template, but ultimately you have it dispersed throughout or and fragmented. So part of each new DNA that is produced is part old, part new, in a fragmented manner, as you see here. Then the semi-conservative replication model or theory suggests that the DNA, parental DNA that you see here in blue, is used as a template. And then what you ultimately have is one strand that is old and the other strand that is new. Keep in mind, we talked about DNA in our last chapter. DNA is a double-stranded molecule. And so ultimately, once there's two strands that make up a molecule of DNA. Okay, so these are the three models that were proposed for how DNA replicates. In order to test this concept, Meselson and Stahl did an experiment to understand how DNA replicates. And I want to make sure I point out to you that this is an extremely important model. This is probably one of a foundational model that is used in genetics. So make sure that you are aware of this concept, especially for those that are taking the MCAT or going to graduate school, as this is going to come back up for you. So I want to use this diagram to explain this concept. So there are two, in the Meselson and Stahl experiment, there are two isotopes of nitrogen that were used. The N14, which is the common form, and the N15, which is the rare form, also known as the heavy form. Okay, and so E. coli was grown up in the N15 media first, so the heavy or the rare form. Then after growing up after one cycle, it was then transferred into N14, which is the common form of nitrogen. The culture E. coli were then subjected to equilibrium density. That's what you see here. So put in a column, okay, which has a heavy salt solution, and allowed to undergo centrifugation, which applies a high centrifugal force, causing separation of the components within the tube. Because of the density gradient that is allowed to form or develop, it separates out the DNA based off of the isotope. So the heavy versus the light, N14 being the light, N15 being the heavy. And so from this experiment, they were able to then determine the model of replication. Okay, so this was the question that was posed for this experiment. Which model of DNA replication, conservative, dispersive, or semi-conservative, applies to E. coli? Now I want you, as we're getting ready to explain this experiment, I want you to think about the concept of what the theory of these three different models suggests, as I previously mentioned. Okay, so as I just showed you all the centrifugation step and using the different, the density gradient to separate out the molecules of DNA, this is exactly what was done. Okay, the N15, E. coli grown in N15, and then after one round of replication, it is moved to N14 and subsequently after. When it is run on a centrifugation or high centrifugation, you see that there are going to be different bands that show up that correspond to the weight of the DNA that is being centrifuged. And you'll see here initially you have one band that shows at the N15, which is all heavy nitrogen. And then here in the second round, you have an intermediate band. What that intermediate band shows is that there is a molecule of DNA that is not 100% N14 and it's not 100% N15. That means it's containing both. At this stage here, one could assume that the DNA can either be dispersive or semi-conservative, right? So then another round of replication, another round of centrifugation occurs again after you have the N14, subsequent N14 growth of the E. coli. And now you have two bands. You have that intermediate band, and then you have the light band, N14. And this continued thereafter, okay, with this intermediate band and then the N14 band. Given the results from this study, it was concluded that DNA replication occurs in a semi-conservative manner. The reason why it could not be dispersive is because dispersive would suggest that every round would always show an intermediate band, because there's fragmented DNA of old and new. But that is not what we see in this study. We see an intermediate band and then over time a higher N14 band, which suggests semi-conservative replication. Okay. So given that information, let's go into how DNA replication takes place in a semi-conservative manner. Okay, so let's talk about the different modes or types of replication. So we have replicons. A replicon is a segment of DNA that undergoes replication. Okay. Each replicon, which is a unit of replication, has an origin of replication. You also have the theta replication. Theta replication occurs because you have circular DNA that has an origin of replication and it forms a replication fork, and it's usually bidirectional, meaning that the DNA is going to replicate in two directions. Okay. And the reason why it is called theta replication is because when you have the structure that is undergoing replication and it gets to what we call an intermediate stage or structure, it looks like the Greek letter theta. That is how the term theta replication was derived. And then you have rolling circle replication. Rolling circle replication is initiated by a break in one of the nucleotide strands, which exposes that 3' hydroxyl group and the 5' phosphate group. So once that happens, you have the new nucleotides that are then added to the 3' end of the broken strand using the inner or the unbroken strand as a template. Once the new nucleotides are added to the 3' end, the 5' end of the broken strand is displaced from the template and it rolls out like a thread being pulled off of a spool. This is how the term rolling circle replication was derived. And you see this with the F factor of E. coli or viruses, and it has a single origin of replication. So just to give you a pictorial representation of theta, this would be an example where you have the intermediate stage, and this is where you get the theta replication from. And then this is your pictorial representation of the rolling circle replication, right? That initial nick on one strand, and then you have that rolling out, okay, forming that secondary structure, the secondary strand. All right. So all of those prior examples were examples of replication for prokaryotic like cells, bacteria, okay, that have circular DNA. But there are instances where DNA replication occurs in eukaryotic cells and their DNA is linear, not circular. So that provides another level of complication that has to be addressed, okay? In linear eukaryotic replication, there are thousands of origins, okay? Thousands of origins of replication. A typical replicon is about 200 to 300,000 base pairs in length. So let me show you a diagram of that, as you see here. So there are multiple origins of replication. And I'll even go forward to tell you, if there were not multiple origins of replication, replication would take days to occur. So this is why in eukaryotic cells, there are multiple origins of replication to ensure that replication can occur at a more faster rate and occurs more efficiently. Okay, so how does this work? You have the DNA that unwinds and produces a replication bubble, which you see here. Then once the bubble is formed, it allows for DNA synthesis or replication. Know that that term can be used interchangeably, the DNA synthesis and DNA replication. So it can take place on both strands. At the end of each bubble, you have what we call a replication fork. This is a replication fork here. And eventually, as replication continues, you're going to have the replication bubbles that eventually will run into each other, as you see here. Okay? And the end result is producing two linear DNA molecules that are identical. All right, so here we have just some characteristics. I would strongly suggest that you use this table 12.2 as a tool to study, understanding the differences between the three models of replication, theta, rolling circle, and linear. And it gives you an understanding of the origin of the DNA. So what does the morphology of the DNA look like? Is there strand breakage? Are there replicons? And if so, how many? And then what is the direction of the replication bubble when replication is occurring? All right, so we've talked about the different models of replication, but be clear that replication occurs in a semi-conservative manner, Okay, specifically now we're talking about eukaryotic replication, linear eukaryotic replication, and there are some requirements for replication to occur. One, there has to be a template strand. That is the strand that is used as a template to make daughter or new DNA. Okay, you need raw materials. Those raw materials are going to be nucleotides, because remember, if you're creating a new strand, you need the materials to create that new strand, which are nucleotides. DNA is composed of nucleotides. And then to carry out the enzymatic functions of this process, because it is an enzymatic reaction, you need the enzymes and other proteins to help carry this process out. So let's visually look at the requirements in action. So here you have your template strand in gray. Okay, here are your this is a nucleotide, a sugar, phosphate, and a base. Okay, sugar, ribose, phosphate, and a base. That is your nucleotide. Okay, to show you a little bit up close, here's your nucleus, your phosphates, there's your sugar, and then your base. These nucleotides are added to the growing strand in a 5 to 3' manner. Remember in last chapter I talked about what that 5' and 3' means. It's referring to the carbons on the ribose sugar. So here you have the 3' carbon on the ribose that is available, and so an incoming free nucleotide can be added to that growing chain. Now there are a couple of requirements or things that have to occur. One, whatever base is added has to be complement to the base that is on the template strand. So in this case, on the template strand, the next base that is free is a guanine. That means a cytosine molecule has to be added to the growing strand, because it has to be complement to the template strand. Second, the initial bond that is formed in this growing strand and the new strand is a phosphodiester bond, and it forms between the sugar and the phosphates. And then lastly, you have the reaction that occurs between the cytosine and the guanine, which we've talked about is the hydrogen bonds that form, and between those two are 3. But you also have what we call other electrostatic forces that help to contribute the coming together of these two strands in a bond-like manner. But ultimately, those are the 3 major things that you need to keep in mind when you have a growing chain that occurs in replication. Okay, so this is just what I summarized for you and what's happening in DNA replication, specifically for linear eukaryotic replication. One other thing that I want to point out is the enzyme that is helping to do the work, and that is DNA polymerase. DNA polymerase is adding those nucleotides to the 3' end of that growing strand. Okay, now because there are 2 strands of DNA, you're going to have continuous and discontinuous replication. Okay, what I mean by continuous and discontinuous replication, there are 2 strands that make up this DNA, this molecule of DNA. But remember, the enzyme DNA polymerase can only synthesize DNA in a 5 to 3' manner. However, the direction of the 2 strands are opposite of each other. So one strand is going to be 3 to 5. That means it will allow for the preferred direction of the 5 to 3' manner of synthesis. However, the opposite strand is 5 to 3. So it's going to occur or cause some potential issues with how DNA replication can occur given the DNA polymerase enzyme. And so because the 2 strands are opposite or have different polarities, one strand is going to be replicated in a continuous manner, and the opposite strand is going to be replicated in a discontinuous manner. Okay, so this is why we also say that DNA replication is not just semi-conservative, it is also semi-discontinuous. Okay, semi-discontinuous. This helps or brings up the concept of the leading and lagging strands. And so I want to just use a diagram to help explain that to you. So remember, we talked about the 2 strands of the the different polarities of the two strands of DNA, one strand being 3 to 5, the other being 5 to 3. Given that their directions, it also is associated with the origin or the direction in which DNA polymerase can synthesize. So the 3 to 5 prime strand here can be synthesized in a 5 to 3 prime manner. So you have a continuous replication. So we denote this strand as the leading strand. Okay, it leads the way in replication, and it occurs at a faster rate than the opposing strand. The opposing strand is going to be the strand that goes in the direction of 5 to 3 prime. And in that strand direction, it is going to have the opposite. Okay, so it is replicated in a discontinuous manner. Therefore, it lags behind in time. This is how it gets the term lagging strand. But what's interesting about this is Dr. Okazaki, who discovered this, found that looking under a microscope, the lagging strand had fragmented or segments of DNA or segmented fragments, okay? And it was occurring or lagging behind in time in comparison to the opposite strand. So this is called, these fragments that he saw are called Okazaki fragments, okay? Okazaki fragments. And this lagging strand that contains these Okazaki fragments is associated with DNA replication or the synthesis of DNA in a discontinuous manner. Discontinuous manner. Now, just to put something into perspective, is because there are fragmented strands of DNA, they have to be joined together, and the enzyme that's going to help do that is DNA ligase. DNA ligase. And the ligase is an enzyme that ligates or joins together those fragmented sections. So here you have the theta model, and diagram B you have the rolling circle model, and then in C you have the linear replication or eukaryotic replication, which has the leading and lagging strand, or is showing a semi-discontinuous manner of replication for both strands. Regardless of the different types of or the different models of replication, all produce DNA molecules by semi-conservative replication. Okay. All right. So let's go into bacterial replication. Okay. And I want you to understand that when we're talking about replication, I've provided you some foundation just to understand how it works. All of the replication require enzymes to carry out this process. Enzymes and proteins. Okay. Enzymes and proteins. So there are 3 stages that we can divide replication into, whether it's prokaryotic cells or eukaryotic cells. That's initiation, elongation, and termination. With initiation for a bacterial cell, okay, you have your site of origin. You also have, excuse me, you have your site of origin. You have an initiator protein called an E. coli called DNA A. And this initiator protein is essential for binding to the origin of replication to help initiate the process of replication. And then you have a melting or unwinding of the DNA strands so that the machinery can get access to both strands. Okay. What helps? There are a series of proteins that help with this unwinding portion. You have initiator proteins, the DNA A. You have DNA helicase, which is an enzyme that helps to denature the 2 strands that are being held together by those hydrogen and electrostatic forces. You also have single-stranded binding proteins, or SSBs, and these single-stranded binding proteins actually line the backbone of the 2 strands to prevent re-annealing or the rejoining of these 2 strands together. And then you have DNA gyrase, which is a type of topoisomerase, and that enzyme helps with the relieving the tension that can occur from supercoiling. So let's look at a pictorial example of that. So here you have your initiator proteins, okay, that bind to the ORC, or the origin of replication. Then you have your other molecules that help to unwind the DNA, a short portion of the DNA, as you see here, to unwind it, causing like a bubble to form. And this allows for the machinery to get access to both strands. Once you have that unwinding and the short stretch of DNA that unwinds and opens up, then you have helicase that can come in and bind and begin to help form that replication fork to continue opening up that bubble. But you also have those small little dots you see are the single-stranded binding proteins, and they're going to line to the backbone of DNA and help prevent the re-annealing or the reforming of the two strands. Okay, and just a more close-up of just DNA helicase and gyrase. So here is DNA helicase right here, and as you can see, it is at the front of the replication fork, helping to open up the bubble and allow for the movement of this replication fork to occur where active replication is occurring. And then you have DNA gyrase, which is occurring in front of the bubbles on either side, and it's helping to relieve the tension. I want you to think about if you had a lanyard and you were twisting the lanyard to where it is, it has coils in it. If you have the lanyard and you begin to try to open up a small bubble in the lanyard, one side of the lanyard is going to have more tension than another, and the reason is because you're causing tension to form on one end. And what happens with DNA is if you don't have DNA gyrase, which is a type of topoisomerase, to help relieve that tension, you can actually cause DNA breaks in the DNA. So it is imperative that DNA gyrase is a part of this process to prevent DNA damage. All right, the next stage is elongation. This is the stage that we've already talked about, and this is where you just have the chain elongation. This is where free nucleotide, free bases are added to a growing strand from the 3' end, okay, the free hydroxyl group, and they occur about 10 to 12 nucleotides in length. And you also have primers that are added now. Okay, so you have the primers that are about 10 to 12 nucleotides in length that are added to the 3' hydroxyl end. And you also have primase. Primase is a type of RNA polymerase that adds down, it's the enzyme that adds the actual primers to the DNA. And from the primers, the machinery uses the primers as an anchor to add on to create what we call a growing strand. So let me show you an example of that. So here is your origin of replication. You see your primase, which is laying down in green, the primer. It's an RNA-based primer. You have your helicase, which occurs on either end of the replication bubble, and then your gyrase on either side. DNA begins with DNA polymerase, start from the primers to adding on what we call chain elongation, and you can see there's the leading strand. Replication is occurring in a 5' to 3' manner, and then you have the lagging strand, which is going in the opposite direction. So we're seeing here clearly that there are two strands. Their DNA is occurring in a bidirectional manner, and it is semi-conservative. We have our leading and our lagging strand, and we see the different roles of these enzymes and proteins in helping to carry out the process of DNA synthesis. Now with elongation, okay, as I've kind of already mentioned to you, I would definitely mark this slide. These steps here are important for efficient and effective replication, specifically the elongation step. And this step of elongation is carried out by DNA polymerase 3. Let me be clear, there are multiple DNA polymerases, okay? There are multiple DNA polymerases, but elongation is carried out by DNA polymerase 3, primarily by DNA polymerase 3. DNA polymerase 1 is responsible for removing the primer, okay? Excuse me. And then you have termination. And we're specifically talking about bacteria at this point. Termination is thought to occur when the replication forks meet. So once two replication forks meet up, it is thought to stimulate or initiate the process of termination. Or secondly, it is thought to be associated with a enzyme what we call a terminator protein to help stimulate the process of termination. Okay, so here are the main polymerases, which are 5N E. coli. You have DNA polymerase 1 through DNA polymerase 5. A couple of things to point out as you're studying this table. Okay, there is different types of activity in terms of the polarity of the strand. You have the 5 to 3 prime activity and the 3 to 5 prime activity. 5 to 3 prime activity is associated with synthesis. But you also have 3 to 5 prime exonuclease activity, which is associated with the removal of bases and also repair mechanisms. So I want you to pay attention to the exonuclease activity because that's going to be essential in the DNA repair. Okay, so here's a list of what I kind of already explained of how this process works of replication, and it's for all of the different polymerases. Now this is important because Arthur Kornberg, who was initially responsible for discovering DNA polymerase, did studies where they caused a mutation in one of the polymerases and found that even when the polymerase that they thought was the sole polymerase was responsible for carrying out replication, it still occurred. This suggested to them that there are multiple DNA polymerases, and we know now that there are 5 in E. coli that carry out the process of replication. Okay, so another thing that I want to point out to you is what happens when replication is finished and you still have this RNA primer that is on the new strand of DNA. That RNA primer has to be replaced. So this is why you have exonuclease activity, because that RNA-based primer, which is just a 10 to 12 nucleotides that are RNA-based, have to be removed. And then the gap has to be filled in by DNA polymerase 1. And then the gap or the nick that's present between where the primer ends and where the strand of DNA was synthesized at has to be sealed. And that sealing of that nick, that space between the two, which you see right here, is done by DNA ligase. DNA ligase is an enzyme that ligates, meaning join together these two, this nick or these two spaces, and it's between the 5' end of one ribose and the 3' end of another. Table 12.4 is a very helpful table because it's telling you and listing out all of the components required for replication in bacterial cells and their function. Okay, we've already gone over these, but I want to point this out so that you can use this as a study tool to study replication. All right, so let's go into a little bit of the fidelity of DNA replication, okay? One of the ways that you can have increased fidelity of replication is through proofreading, okay? Remember, in the very beginning, we talked about errors in replication is inevitable. It's going to happen. Whenever you're adding in bases at such a fast rate, you're bound to have some type of error that occurs, whether it's the wrong base being added or the base being twisted in such a way, which we call like a tautomeric shift. It's inevitable. And so DNA polymerase 1 is essential in going back and proofreading, so going back over the DNA that was just added to ensure that there are no incorrectly paired bases. And so this is an important part of DNA replication is the proofreading aspect. Okay. You also have mismatch repair. So not only will it look for incorrectly paired nucleotides, but also try to correct errors that are present after replication is occurred. So let's say there are some errors that are overlooked even after the proofreading. Mismatch repair will be activated to make sure that there are no errors. So to answer that initial question that I asked you is how can we have so many errors that can occur, but we always don't see the physical manifestation of those errors? It is because we have an innate response called DNA proofreading that helps to repair as many of those errors as possible. All right. So with eukaryotic replication, okay, you have what we call autonomously replicating sequences, or Rs. And these are DNA sequences that confer the ability to replicate, and they contain an origin of replication. Okay, they're about 100 to 120 base pairs. And we also have ORC. This is an origin recognition complex. And this ORC binds to the Rs to help initiate DNA replication. Okay, so this is important in eukaryotic DNA replication. Now, why is the ORC important? The ORC recruits and loads DNA helicase, and we already know the function and the importance of helicase. It helps to denature those hydrogen bonds that keep the two strands together. And so this helicase in the ORC is extremely important. Now, a clear difference between eukaryotic cells and prokaryotic cells, specifically bacterial cells, when it comes to initiation is eukaryotic cells don't necessarily need an initiator protein. So DNA A is not a requirement for eukaryotic cells. However, eukaryotic cells do require what we call licensing factors. Okay? Licensing factors give the approval to initiate replication. And these licensing factors are associated with the cell cycle. Okay, they are specifically with those checkpoints. And so these licensing factors are made prior to DNA replication in the S phase. Okay, so remember in the cell cycle you have G1, S, G2, and M. These licensing factors are made in G1 phase. And the production of these licensing factors, which are proteins in G1, ensure that when the cell gets to the S phase to activate the synthesis of DNA, these proteins are produced and ready to begin the process. Okay? The association of these licensing factors is the ORC is going to attach to the origin of replication. And remember, there are multiple. Okay? And they combined with these licensing factors to form what we call an MCM, mini chromosome maintenance. Okay, mini chromosome maintenance. One of the complexes or proteins involved with that is called the MCM27. Okay, and they bind to the origin. Okay, and this also comes back into play in the S phase. Okay, because it's going to initiate the coming together, the coming together of multiple or cofactors to form an active complex with helicase to help unwind the DNA. Okay, so these licensing factors are very important. Another clear difference with eukaryotic cells, and let me say this, you still have the initiation, elongation, and termination, those three stages for replication. Okay, elongation is the same for the most part in terms of just the adding of the free nucleotides to a growing strand. However, I want to talk about some differences so that you understand that there are differences between the two, but the foundation of replication is the same. It still occurs in a semi-conservative manner. You still have a leading and lagging strand. However, in eukaryotic cells, there are at least 15 different polymerases. Okay, and one would assume that this would be the case given the complexity of eukaryotic cells. Okay, there are 15 different polymerases. Okay, the three polymerases listed at the top are those that carry out nuclear DNA replication. Okay, remember there are other organelles that occur within a eukaryotic cell, and so those organelles have their own polymerases as well. But alpha, delta, and epsilon are the main three polymerases that are involved in nuclear DNA replication. So pay attention to learning this table as well. Okay. With eukaryotic cells, another difference between eukaryotic cells and prokaryotic cells in terms of replication and its complexity is histone proteins. Eukaryotic cells have linear DNA, and so you have to have the condensing of this DNA, and it is condensed by the presence of histones. We talked about this in a prior chapter. Okay, nucleosomes are made up of the histones proteins, N2A, N2B, N3, N4. Okay, H2A, H2B, H3, H4. So you have this octamer of histone proteins along with H1 and other proteins that allow for DNA replication to be condensed. Okay, so not only do we have to consider the complexity of the decondensing, the disassembling of the nucleosomes and histone proteins and the reassembling of them, but how does this affect replication? Okay, because these histone proteins, as you can see here, you see this is an example of nucleosome assembly right here, right here. This also is another layer of complexity that contributes to the timing or the delay that we see in replication of a eukaryotic cell versus a prokaryotic cell, because the timing is significantly different. Okay. So just briefly, we won't stay on this long because I want you to make sure you go back and read through this and try to piece together your interpretation of this in your own words. But what the question is, what happens to histones in DNA replication? So that was the question for this experiment. And so ultimately they did a similar concept as before of growing the cells in different medium, the heavy versus light isotopes, specifically for amino acids. And they were able to see the differences during applying centrifugation. And from their experiment, they were able to show that after DNA replication, the new reassembled octamers are random of old and new. So ultimately, after the cell disassembles the histones, okay, once it reassembles the DNA after it's replicated, you have a complex of old and new histones that randomly form these octamers to allow the nucleosomes to form and to allow the recondensing of the DNA. Okay. Another concept that we have to talk about is the telomeres. Okay. The telomeres. Now, this is one of the factors that are unique to eukaryotic cells, and that is the end of the linear chromosomes. Okay. And also, I want to point out too the location. Okay, and eukaryotic cells, because they have membrane-bound organelles. When we're talking about nuclear DNA, replication takes place in the nucleus. In prokaryotic cells, remember there's no membrane-bound nucleus, so replication occurs in the nucleoid region, a dense region in the cytoplasm. So the replication of the ends of the chromosomes in eukaryotic cells present a problem, okay? Because remember, there are primers that are removed after each round of replication, and so if you're not careful, the ends of the chromosomes, as you can see, can get shorter and shorter, okay? So it leaves, once the primers are removed, it leaves a gap that is not replicated, okay? And so it is important that telomeres, and this was a study that was done to understand this even more, is telomeres have to be added, and telomeres are these small tandemly repeated sequences that are added to the ends of the chromosomes. And let me just kind of scroll this so you all can see. The tandemly repeated sequence, let's zoom in a little bit so you all can see that, okay, has this sequence of G-rich repeated sequence, TTAGGGG, okay, that is added on by an enzyme called telomerase. Okay, so telomerase is an enzyme that adds on the ends of the chromosomes to fix that gap issue that it occurs on the ends of these linear chromosomes. And it's important that it is repaired or is added on, because if it would not, then the cell would be truncated for apoptosis or stimulated for apoptosis. And there have been many studies that show telomerase activity, and the ends of chromosomes, i.e. telomeres, are associated with the age of a cell. Okay. So we're going to I'll go to this and then we'll briefly, we're going to end here. But so outside of replication occurring, okay, you also have what's called recombination, okay, specifically homologous recombination. And we talked about part of this earlier in the semester. Homologous recombination is the exchange between homologous DNA molecules during crossing over. And we know crossing over takes place during the cell cycle, specifically prophase, okay? But we also have instances where a form of recombination has to occur when there's DNA damage. Part of that can be attributed to what we call the Holiday junction and associated with a single strand break, okay? But you can also have double strand breaks, which are much more severe, and those are a way that you can have repair as well through Holiday junction for through recombination. Okay, and so that's what you can see here. Okay, here's homologous chromosomes that they align, and the single strand breaks occurs at the same position. You have the free end of each of the broken strands that migrate to the other DNA right here. And as they do that, they invade the strands that join together, and they create their invading of each other. The opposing strands form this Holiday junction. Okay, and once that forms, it begins to displace the original complementary strand. Once you have active there's recombination that's occurring, it forms what we call a heteroduplex DNA, meaning that each molecule is hetero. It's part, now I want to say old and new, but it is DNA from opposing strands of DNA. So it's not homologous technically anymore because it's containing part of the opposite strand. And so this is just showing you the different planes or view of the DNA post the recombination that occurs. And you can see here, you can have non-crossover recombinants and crossover recombinants. But what this is doing, this Holiday model that was proposed predicts non-crossover and crossover recombinants of DNA, and it depends on whether the cleavage is in the horizontal plane or the vertical plane. So just how the DNA is cleaved. Okay. The double strand break model of recombination occurs when you have a double strand break and there's no template that can be used to repair it. So think about if you have a break in the DNA due to, let's say, an issue or error in replication, but both strands are broken. There's no way to use one strand as a template to repair the other. And so when you have a double strand break, you have this model, this Holiday junction that forms to ultimately form that heteroduplex to try to repair both strands of DNA. Oftentimes with a double strand break, you can have the loss of genetic information. All right, so this is chapter 12 on DNA replication and recombination.